mic.
मेरे में तो नहीं दिख रहा, पीपल 92 देखिए इसमें लिस्ट में होगा।
I request all the participants to please mute your mics.
Okay, Harke, madam, continue.
Yes. Now I kindly request our principal sir to deliver the inaugural address and set the tone for this academic journey, sir. Sir, please, sir.
Okay. A very warm and pleasant morning to one and all. It gives me immense pleasure to welcome you all to the 1-week International Online Faculty Development Program on Cutting Edge Perspectives in Pharmacy, Translating Molecules into Medicine, organized by VV Institute of Pharmaceutical Sciences, Gudlavalleru, in association with Association of Pharmaceutical Teachers of India, APTA Central Bangalore, from 13th to 18th October 2025. The Association of Pharmaceutical Teachers of India, APTA, established in 1966 by the pioneers of pharmacy education, such as Professor M.L. Sharaf, Professor G.P. Srivastava, and others have played a pivotal role in fostering communication and promoting excellence in pharmacy education across the nation. Through its well-defined mission and objectives, APTA provides a common platform for uniting pharmacy educators and strengthening the academic collaboration. Celebrating 57 years of dedicated service, APTA today boasts over 15,000 life members, along with numerous associate and student members from across India. Its diverse activities promote innovation in teaching methodologies, computer-aided drug design, and industry-academia interactions, while fostering the collaboration with hospitals, regulatory bodies, and community pharmacies to advance pharmaceutical education and practice. We are truly honored to have with us today our chief guest and my good friend and patron of this particular FDP program, Dr. Ramandan Garu, Secretary, APTA Bangalore. Other esteemed patrons of this FTP, Dr. Milind Janrav,
Sir, unmute yourself.
Sir, principal sir. Sir, you have to unmute, sir.
Okay.
Okay, sir. Thank you, sir.
Okay. A very warm and pleasant morning to everyone. It gives me immense pleasure to welcome you all to the 1-week International Online Faculty Development Program on Cutting Edge Perspectives in Pharmacy, Translating Molecules into Medicine, organized by VV Institute of Pharmaceutical Sciences, Gudlavalleru, in association with the Association of Pharmaceutical Teachers of India, APTA Central Bangalore, from 13th to 18th December, October 2025. The Association of Pharmaceutical Teachers of India, popularly called it as APTA, it was established in the year 1966 by the pioneers of the pharmacy education, such as father of the pharmacy, Professor M.L. Sharaf, Professor G.P. Srivastava, and others have played a pivotal role in fostering communication and promoting excellence in pharmacy education across the nation. Through its well-defined mission and objectives, APTA provides a common platform for united pharmacy educators and along with the associate and student members from across India. Its diverse activities promote associate and student members across the India, and it includes the teaching methodologies, computer-aided drug design, and industry academia interactions, while fostering collaborations with hospitals, regulatory bodies, and community pharmacies to advance pharmaceutical education and practice. We are truly honored to have with us today our chief guest and patron, my good friend, Dr. Ramandan Garu, Secretary, APTA Central Bangalore, our esteemed patrons of this FTP, Dr. Milind J. Umekar Garu, National President, APTA Bangalore, Dr. M. Venkatramana Garu, Vice President, Southern Region, APTA Bangalore. It is also our privilege to warmly welcome the today's eminent speaker, Dr. Preeti Vyas Garu, Department of Anesthesiology and Critical Care Medicine, Johns Hopkins University School of Medicine, Baltimore, USA. along with other distinguished faculty participants from various reputed pharmacy institutions across the country. Allow me to briefly introduce our institution. The VV Institute of Pharmaceutical Sciences, popularly called it as VVIPS, was established in the year 2010 under the aegis of the AANM and VVRS Education Society, Gudlavalledu, Krishna District, Andhra Pradesh. We offer B.Pharm, M.Pharm, Pharmaceutical Analysis and PhD programs. The college is recognized by the Pharmacy Council of India, New Delhi, and affiliated with JNTUK, Kakinada. And our college is NAAC accredited and ISO 9001-2015 certified institution, and has established itself as one of the premier institutions for pharmacy education in the state of Andhra Pradesh. We take immense pride in hosting the 6-day FDP featuring with 6 distinguished speakers from across the globe. The program aims to provide an in-depth understanding of the latest advancements and innovations in pharmaceutical sciences, holding the gap between molecular discovery and therapeutic application. It will highlight the role of modern drug design, formulation technologies, regulatory insights, and translation research. Experts from academia and industry will discuss emerging areas such as AI-driven drug discovery, nanomedicine, targeted delivery systems, and personalized therapies. This FDP seeks to empower faculty members and researchers with contemporary knowledge and practical approaches to enhance teaching, research, and innovation in the pharmaceutical domain. I am confident that the insightful lectures, discussions, and interactions planned throughout the week will inspire participants to integrate these emerging trends into their academic and research pursuits, fostering excellence and innovation. At this juncture, I extend my heartfelt gratitude to the visionary leadership of our management for their constant encouragement and unwavering support, which continues to drive excellence at our institution. A special note of appreciation goes to Ms. M. Harika, Assistant Professor, Department of Pharmaceutics and convener of this FTP program, for her dedication and initiative in organizing this program. And also to Dr. Sheikh Amina Bigadu, FDP Coordinator, VVIPS, for her meticulous planning and coordination. I am delighted to share that this FDP has received an overwhelming response, with more than 500 faculty members registered from over 250 esteemed pharmacy colleges across India. Your enthusiastic participation reflects your commitment and also in advancing the pharmaceutical sciences. On this first day, we are privileged to begin with Dr. Preeti Vyas' lecture on From Discovery to Delivery, the Translation Pathway in CNS Drug Development. The session will provide an insightful overview of the complex journey involved in bringing the central nervous system, popularly CNS therapeutics, from the laboratory bench to clinical application. It will emphasize the unique challenges that are observed associated with the CNS drug discovery, including the blood-brain barrier permeability, target validation, pharmacokinetics, and safety profiling. This talk will also explore recent advances in neuropharmacology, biomarker identification, and translation models that bridge preclinical and clinical research. By integrating cutting-edge strategies in medicinal chemistry, formulation design, and regulatory perspectives, the session aims to offer participants a comprehensive understanding of the multidisciplinary efforts required to successfully translate CNS molecules into effective therapeutic agents. I am certain that the session will be both enriching and inspiring for all participants. I take this opportunity to express my sincere thanks to the Association of Pharmaceutical Teachers of India, APTA Central Bangalore, for partnering with us in organizing this FTP program. My deep appreciation also goes to Dr. Milind Chandra Umekar, Dr. M. Venkatramana, and Dr. Ramandang for their continued support in conducting this program for the benefit of the teaching fraternity. And we're also thankful to Ms. Preeti Vyas. She's working as one of the premier world top university in USA. And today we are going to happy to inaugurate this FDP with particular Madam's lecture. Thank you very much, Madam. I wish this faculty for development program great success for the academic benefit of the teaching fraternity members. And I hope it provides a highly rewarding learning experience to all the participants. Thank you one and all for being a part of this academic journey. Now I invite you, Ms. M. Harika Garu, convener of this FTP program, to proceed with today's program. Over to Harika Garu.
Thank you, sir. Thank you, sir, for your enlightened strength and words, which gives the strength for this FDP program. Now, I would like to invite Dr. D. Ramanadang, sir, Secretary at APTI Central Bangalore, to have a few words about the FDP. Sir, please, sir.
Yeah, thank you, madam. Thank you, Lakshman sir, for inviting me. And now this FDP, as you see, this has become a conventional part of teaching learning systems, and that too making it international shows the magnitude of your institute, VV Institute of Pharmaceutical Sciences, that you are connecting with the international scenario of drug development, drug discovery. These screening components are very essential for us teachers. And when we take it forward, obviously students also benefit. Along with that, I have seen that Lakshmana sir does a lot of hands-on training, and the institution's chief patron, Dr. Nageshwar Rao, and our Srinivasa Rao, we really congratulate you to have such a strong team which are scientific bound. Otherwise, at the principal position, what all Lakshman sir is contributing is not easy, because it's a more of administrative component, and he's always into science and very, very alert in all the aspects, whether it is of APTI or any professional activity. So fortunate for the people who are working with him. And our Ramakrishna sir also, as they've mentioned as co-secretary, we really pass on the respectful regards. My friend Milind sir, who is definitely very, very busy with now IPC. So I'm happy to be connecting with you people on behalf of APTI. Ramanna was here with us yesterday. So continuously, there are different kind of activities happening, which is also teaching learning, which will have a forum of networking in the form of IPC. Convener Harika ji also should be congratulated for connecting with so many resource persons. I was seeing your brochure, and it is very, very impressive that you're seeing to it that there is a total gain for all the participants. And having 500 participants is a big number. So this thing is I think, learning for all of us to see that hybrid mode should continue. We had got a lot of requests for IPC also to have hybrid mode, but that is a protocol bound function. We could not do it. But teachers should attend FDP, especially when your institute is hosting it. This is a knowledge gain. And APTI, as you all know, is always encouraging, always part of such kind of FDPs, which are gaining the knowledge to the young teachers, especially. And a lot of industry also is coming forward. Yesterday, Harish Jain, Atawaji, so many people we connected with, and they says, yes, we are keen to connect with institutes through FDP so that practical knowledge gain can happen. Even demonstration, why not? Even demonstration can have usage of high-end instruments. Since you have analysis specialization in MPharm, can also be one part, Lakshmanji. Please think on it. So hands-on, instead of sending your teachers there, you can have
Sure,
it on
sir.
online also. So that will be a gain. So my suggestions come only to those who are working in that direction. And Lakshman is always very, very close to me in terms of that. So again, once again, I will congratulate you, and I will invite you also to Indian Pharmaceutical Congress at Bangalore. Be part of the bigger event, be part of the panel, networking, and connecting with the scientists. So for your future FDPs, you can invite such people whom you know during this networking process at Bangalore. So good luck for your process of research, and your FDP 1 week is a huge period, but you will do justice. In between, if time permits, I will definitely join and attend, because it is also knowledge gain for me. Thank you for inviting me. Good luck and best wishes.
Thank you, sir. Thank you very much, sir. I'm happy to say that totally 70 members are attending from our college for the APTI IPC, that is 74th IPC at Bangalore, sir.
Very good, very good.
Yes, sir.
Make it the best.
Yes, sir. Thank you, sir.
Thank you, sir, for your continuous support to the institution. And we thank APTI for this association and a wonderful collaboration. And we hope to continue the same work together for many, many years. Now, continuing to our section, now I would request Assistant Mansa Madam, Assistant Professor of BVI IPS, to introduce distinguished speaker, Dr. Preeti ma'am.
Mansa madam, you are not audible.
So, yeah, sorry. Good morning, sir. Sir, now I'm audible, sir.
Oh, you are not audible, Mansa madam. You can continue.
Yes, sir. Good morning, sir. Good morning, everyone. My name is G. Manasa, working as an assistant professor in the Department of Pharmacology at the VVIPS. It's my privilege to introduce today's speaker, Dr. P. Preeti ma'am. Ma'am has postdoctoral research fellow, Department of Anesthesiology and Critical Care Medicine, Johns Hopkins University School of Medicine, Baltimore, USA. Ma'am is postdoctoral research fellow with expertise in Translational Neuroscience, and she holds an MPharm PhD in the pharmacology from Jamia Hamdard, India, where her doctoral research focused on neuroinflammation and epilepsy through dual inhibition of PI3K and mTOR pathways. Ma'am has published numerous high impact articles in SII and Scopus Index journals and contributed to book chapters and has presented her work at international conferences including SFN, MAPS, and ACCM Research Day. Ma'am has mentored undergraduate, masters, and pre college students in laboratory research and thesis projects. She received multiple awards recognizing her research excellence, including the Psychedelic Science Scholarships at the year 2025, Trainee Professional Development Award 2024, and several Best Foster Awards at Johns Hopkins ACCM Research Days at the year 2022 and 2022 to 24. Ma'am was also a DSI Inspire Fellow of at the year of 2016 to 2021 and awarded the Best Research Fellow of at the year 2021 by the Society of Pharmaceutical Educational Research India. Ma'am work bridges preclinical research and translational therapeutics, advancing novel interventions for epilepsy and neurodevelopment disorders. Thank you, ma'am. Thank you.
Thank you so much. Can I share my screen?
Yes, ma'am. Thank you, Miss Mansa, madam, for your introduction. Now, I would like to request Dr. Preeti Vyas, ma'am, to take over this section and enlighten the participant with her valuable insights. Ma'am, please go on, ma'am.
Thank you so much. I'm really excited to be a part of this faculty development program and to interact with so many senior people in the field. Hello, everyone. My name is Preeti, and I work at the Department of Anesthesiology at Hopkins. I think I am introduced in such a long, with such a long introduction, but I'm really excited that I'm here, and I thank VVIPS for having me here today. So this we already talked about a lot, that I have a broad background in developmental research, and I'm really grateful that I've been a part of different good institutions in India. I have also received some industrial training while my stay in India. I did my master's thesis at Daiichi Sankyo, which is a Japanese company, Japanese pharmaceutical company. And from there, I got really interested in drug discovery. And because of my training at Daiichi, I went back to Jamia Hamdard and did my PhD in pharmacology. And during my PhD, I spent good 3.5 years at DRDO DePass, where I did most of my PhD work. And when I completed PhD in the middle of the COVID, I immediately came to the US to the Kennedy Krieger Institute. I was a part of Michael Johnston Center at Kennedy Krieger, as you can see here, maybe. Yeah. And then I joined Johns Hopkins, and I always like to show this picture of this dome because this is like the main Hopkins hospital. And just in case if you want to contact me or connect me with LinkedIn, I also linked my details here. And this is the picture from one of the most recent presentations that I did in Philadelphia. So I wanted to share that, and I'm excited to share some of my data. So today in this presentation, what I'm going to show is a good roadmap of the translational journey that we see for all the neurotherapeutics that comes from preclinical research to clinics and to bedside of the patient. So I think this is something we all are familiar about, that how drug discovery and development happens. But what I'm going to do today is to add some aspects of some like very modern aspects to it, like what is being done currently and what are the changes we need to do, what is the troubleshooting we need to do. And we are also going to see some of my data that I have acquired in past few years. So I included few slides of some experimental data also. And let's see how the drug. Oh, yeah, I think something happened. Okay, let's see how this whole drug discovery process happens. So I think for this particular slide, probably I'm not the best person to talk about because I'm not a medicinal chemist. But the initial stage of any drug discovery process, it begins in a chemistry lab with basic research to understand the disease at a molecular level. Okay, so first, if we want to look at drug discovery process, what we want to look at, we first want to look at the target that we want to like create a molecule that binds to a particular target or specific gene or a protein, right? So target identification is one of the first steps. But what are the problems with target identification? So we know, especially with CNS diseases, most of the diseases are multifactorial. So if for example, I have worked on PI3K during my PhD. So I was inhibiting PI3K, and then I was observing that once I inhibit PI3K to treat epilepsy, some of the other molecules or some other mediators or proteins, their level go up. So there is always like a feedback compensatory mechanism that you inhibit one target, other go up, and the disease still remain. So what is the best way to identify a target and validate it, and then prepare compound, optimize the leads, and how to proceed in this direction that we are going to talk today. So we all know about structure activity relationships probably, and I don't think there is a good way to get into it here, like it's out of the scope for this presentation. But we first identify a pharmacophoric element, and then we, through our structure activity study, we design a generation of different molecules. So we have like a hit series where we have different molecules that probably interact with a particular drug target of interest. Okay, then what we do, out of all those molecules, we select few promising compounds that are called leads. So next, we do the lead optimization. And when we prepare these compounds, we test their biological activity, selectivity, pharmacokinetics, and then we do analysis to confirm their structures, like people do NMR, mass specs, LCMS, and then we repeat the cycles using new data and refine these leads compounds. Now, this is the part that I think I can talk about more. So once we have, so in when I was in industry, the main work that I was doing is I was receiving compounds from chemistry, and I was doing like a good pharmacological screening on them. So what is the first step when you have a compound or have a molecule? What should we do? How should we push it into clinics? So the first step is in vitro efficacy testing. So am I going to test it directly on animals? No. Am I going to use it on cells? No. So what is the first thing that I will do when I receive a compound from chemistry? So first thing I will do is to test it in a cell-free environment. Okay, for example, I have a kinase inhibitor. Let us consider that I received a kinase inhibitor from chemistry department. Now I will check whether this kinase inhibitor is actually inhibiting my enzyme or not, right? So I will create a microenvironment, like an artificial microenvironment, which only has my enzyme and my drug molecule, and maybe some salts like ATP, ADP, MgCl2, which are necessary for the compound to actually act. And I will check the IC50 of my compound, and I will see whether it is active or not in a cell-like environment. And once I have my IC50, then I will proceed towards the cell-based assays. Now, when I say cell-based assays, it can be a cell line, it can be a primary cell, or it can be like an organotypic assay. For example, I'm not sure if everyone is familiar with the cell line, who are probably yes. So we have like particular type of cells that we freeze, and we can passage from one generation to other, from like different passages, different divisions, and we can test our compounds on it. So it's like testing your molecule in a dish, right? So I have like a example of it. For example, there are two-dimensional cell culture, there are spheroid cell cultures and organoid cell cultures that are less common. So two-dimensional cell cultures are commonly used and they are very easy to manipulate. For example, I take some neuronal cell line and I grow my neurons in a dish and I test my compound on it, okay? Now, what the problem is here? The problem is that when you are testing your drug on just neurons, you are not creating like a in vivo environment. Because in the body, what happens? It's not just neurons, right? There are microglia, there are oligodendrocyte, there are different types of cells, astrocytes, then neurons. So you need to study when you put your drug molecule there, what it is doing to the whole circuit, what it is doing to every type of cell and how it is affect, because one cell affect the activity of other, right? So we need to do it in a spheroid or organoid like environment, which is actually closer to the physiology of the body. After doing the cell culture, we do in vivo efficacy studies in animal models. And then where we test the therapeutic effects, biochemistry, physiological effect, is there a dose dependence or not? And then we look for adverse effects and toxicity. I think this is the basics of any drug discovery process. So let us see it with an example. So this is some of my data that is already published. You can find it online also. So what I did here is I isolated the primary neurons from rabbits. So I took like a newborn rabbit and I isolated the neurons using a neuronal isolation protocol, okay? And then I cultured them in a dish. So I was growing my neurons on a cell culture plate. And then when they were grown for 2, 3, 4 weeks, I injured them because I want to create a disease-like environment. So to injure them, I added glutamate, okay? So when I added glutamate, my neurons started to proliferate, and then I added my drug therapy and then waited for 24 hours and fixed them to see how much of drug is actually going inside the neuron. Because we always talk about cell targeting, right? So I wanted to see how much my drug, because this is a nanotherapy, which I was putting as a drug molecule. So I wanted to see how much my nanotherapy is actually going to the neurons, how much it is targeting. So as you can see in the picture, the first column is DAPI, which is the nucleus of the cell. Second one, in the green, you can see the primary neuron that I cultured. And the third one is my drug molecule, okay? So you can see that this GD2 here, it is going exactly inside the neuron. In the last column, you can see everything is overlapping. So this is the kind of study you need to do if you want to specifically look at the targeting. So here it confirmed that my drug molecule is targeting the primary neurons. Then I also wanted to do some mechanistic study to see, okay, so here I know that my drug molecule is going inside a neuron, but how it is going inside is another question, right? So I wanted to see how neurons are uptaking my drug molecule. So we know that there are many receptors on the surface of the cell. There are glucose transporters, there are different receptors. The drug also goes inside through diffusion or passive transport, many things, right? So what I did, one by one, I blocked each of these processes. So as you can see, I'm adding cyclocalicin B, which is a GLUT inhibitor. So it inhibits all the glucose transporters. Then I also added another GLUT inhibitor. Then I added one which is co-transporter inhibitor, and then one which is, somebody said slides are not moving. Is it? Is it for everybody? that the slides are not
Going,
moving?
ma'am. No, ma'am, it's their problem. It is okay, ma'am, with your PPT.
Oh,
Please
okay.
carry on, ma'am. Okay, ma'am.
Thank you. So I wanted to see how my drug molecule is going inside the cell. So what is my idea to show you this data is that when you do any cell culture study in a drug discovery pipeline, you need to see whether you can look at different aspects, like how your drug is going inside the cell. What is the mechanism of it going inside the cell, right? So here I blocked different mechanisms, and I saw that if I block the mechanisms, my drugs do not go in. So that's how I elucidated that this is the mechanism by which it is being uptaken. Like in the third column, you can see here, if you can see my cursor, you can see that if I do not add any inhibitor, I see like a very high signal of my drug. If I add inhibitors, the signal go low. And then I counted using like a machine learning code. I counted how many neurons are taking up my drug. And then this is the quantification. So I said that, okay, maybe my drug is going inside through GLUT transporter because this is significant. So this is the kind of study we can do. And now I know that, okay, my drug is going inside the neuron, but is it showing any efficacy in animal models? So this is one of the animal models that I checked. So because I work on epilepsy, so I induced seizures in mice. Seizures are like when neurons get hyperexcited, they keep firing, they become hyperexcited and they fire action potentials and they start seizing. So we created this kind of animal model. I injected pilocarpine, it is a chemoconvulsant, and I observed, you can see this graph number D, and you can see that my drug is reducing number of seizures. So it is also efficacious. So here I looked at three things. Is it targeting? The mechanism by which it is targeting, and whether it is efficacious or not. So this is like this was just an example to show that, okay, this is how we go forward in for developing neurotherapies. Now I have efficacy. Now I want to do like I will do some toxicology studies, calculate LD50, because we all are from pharma. Probably we all know about how we do lethal, how we calculate lethal dose and everything. So I did not add that. But once you have all of this, how will you use the dose calculation that you did in animal model to know that, okay, if I go clinical, how much I need to give to my healthy volunteers, or what should be in human starting dose of my drug? So to test that, we do the PK/PD correlation. Okay. So for example, I will use like a dose which doesn't have any adverse effect at a particular dose. And then I will use a pharmacologically activated drug, pharmacologically activated dose, I'm sorry. And then you will correlate the PK/PD data, and then you will select, okay, this is using biopharmaceutics compartment modeling. You will select that, okay, this is the dose which should go in human for the first trial. Okay. So that's how we calculate first in human dose. So I'm trying to go forward from chemistry lab to clinical trials. So this probably we all are familiar of, that once you select like a first in human dose, I think one phase is missing from here. First we do microdosing and then we go to phase 1. So phase 1 is when there are like a small group of healthy volunteers, and then we go larger in groups. And then as we go like multicentric trials, and then in phase 4, when the drug is actually reviewed and goes to the market, upon the approval, we do the post-marketing studies. Okay, so this is how this whole thing happens. So after phase 3, I forgot to mention this, that the company which is doing all of this, the industry which is doing all these studies, will file a new drug application to an authority like FDA in US or CDSCO in India, or they will do investigational new drug application. And then all of this data will get reviewed. And based on the review, it can either get approved or rejected. And if it gets approved, it goes into market, and then phase 4 is done. All of this is good, very cool. This is how things happen. But what is the problem with neurotherapies? Like, this is traditionally how drug discovery happened. What makes the CNS very unique? And what is the translational gap? So most of the CNS drugs, which goes from labs to clinics, at least 85% of them, they fail. They don't show good efficacy. So what is the problem? Why it happens? Because first thing, we all know about blood-brain barrier. There are tight junctions and the drugs cannot go in. And because of that, we are making nanotherapies. I also work on one of a very good nano platform called Endrimer Nano Platform. So we try to deliver drugs across the blood-brain barrier, even the drugs that are hydrophilic and are not ready to pass through the blood-brain barriers. We manipulate them into different formulations of small sizes, and then we try to make it work. But still, there is like a disconnect between animal models and actual human pathology of the disease. So what we need to do is we need to focus on target validation, and we need to select like really good animal models to fill this gap. Okay, so this is what we were talking about. Target identification. So what we can do to make the neurotherapies work, we need to have like a target identification using human data. Okay, so when we identify a target initially, we are supposed to use the omics data or some data from postmortem brain studies, because that is closer to human system, right? Instead of using animal data, that is a better way of doing target identification. And then we validate that target using human iPSCs, which means the cells that are derived from patients. That will tell us whether the actual disease phenotype, and will tell us whether the target is actually druggable or not. And then we do high throughput screening and lead identification and everything. And then we look at, we balance the blood-brain barrier penetration, we look at lipophilicity, and then in silico studies, which is computational studies, like using AI or other things. And then we can, before even doing the animal studies, we can predict how toxic or how safe the therapy will be, or whether it will go through blood-brain barrier or not. We can do all these studies in advance, so which will increase the success of the drug molecule in clinical trials. Okay. Another thing is that most of the studies that were done previously were done using two-dimensional cell cultures. Like I previously mentioned, that people were either using neurons or microglia or oligodendrocyte or whatever type of cell they wanted. But the problem remained, because we want to study what will happen, what the drug will do to the system when all these types of cells are present, right? So we should go for more complex CNS in vitro systems, like organoid cultures, spheroid cultures, which are very similar to the actual organ. So it's very interesting. I think 2 or 3 weeks ago, I met somebody working in a lab, and they have like a organoid for brain, which is very interesting. And they had like they sequenced it, and they showed me the sequencing, and they were like this. They told me that, oh, see, the sequencing is gene sequencing is very close to the actual human brain. So this is like a very good model to study. In a plate, you can study what is actually happening to the human brain. So that kind of studies will increase the success in the that kind in the clinics, maybe. Okay, so another thing that we can do is to use humanized mouse models. Now, what are humanized mouse models? Just as a revision, we can talk about it here. Let us consider I'm using like a mouse that has like a mouse receptor, okay? And my drug will bind, when I test my therapy there, my drug will bind to a mouse receptor, and then I will see the efficacy. But mouse receptors are not present in human. Human receptors are present in human, right? So I need to use some humanized mouse model that has the receptor which is close to the human receptor. For example, we use like, let us take one receptor like GLP-1. So I want to use a humanized mouse model that has human GLP-1 receptor present in it, so that when I do my drug testing in that mouse, I'm closer to human system. And then we can also do some pharmacokinetics and pharmacodynamic studies, which means like, okay, how much free drug concentration should reach brain parenchyma to show some therapeutic effects. So in mice, what we do, we implant cannula in the brain, and then we keep taking like some fluids from the brain, and we check that, okay, this is the concentration of some neurotransmitter or protein in the fluid over time. So that process is called microdialysis. So you can do microdialysis in mice and rats. And also, you can also look at like cerebrospinal fluid, some imaging biomarker, like you can do MRI, you can do PET scanning, things like that. So these translational biomarkers are very important. So what are the other translational endpoints? To demonstrate that the drug is actually binding to the target. Okay, so you can do PET imaging to show that my drug is going to the target, and then you can do MRI and to see what is the structural and functional changes in the animal. And you can also do, like in clinical studies, we do human PET, okay, positron emission tomography. And then in mouse studies, we also do PET imaging to see like the homology between human and mouse or rabbit studies. You can also do some mass spec assays on rodent cerebrospinal fluid and plasma samples, clinical samples, and you can use some computational models to correlate in vitro data to in vivo data and predict the human doses. Okay, so what are the takeaways? We need to focus on target validation. We need to integrate all the biomarkers across all the stages of drug discovery. And then we need to use better models, like we need to use iPSCs, organoids, spheroids, all these models. And then we need to do a good PK/PD correlation in the brain to make the therapies a success. Okay. So what are the emerging technologies that people use? People these days, we do gene therapies, we do microRNA, siRNA, and antisense oligonucleotides. Now, what are antisense oligonucleotides, ASOs? Just to have like, just for you to have a sense of what they are like short pieces of DNA. Okay. And these short pieces of DNA, they target specific, what they do. So we know that every gene, it produces mRNA, right? So they bind to the mRNA and then they degrade it. So this is how they are silencing a particular gene. So this kind of neurotherapies, if we use, there are more chances of success. Or targeted therapies like dendrimers. Like I told you, I work with dendrimers. And then we can use AI or machine learning target identification, and we can use personalized medicines and stuff like that. So I also wanted to show some of my data. Maybe I'll take 15 minutes more. This data I wanted to show just so that we correlate all of this as like an example. I am okay. Maybe somebody else took a control of my presentation. Okay, so as I already discussed, I worked a lot in pediatric epilepsy in children. So I worked on a seizure model, which is a model of hypoxic ischemic encephalopathy, which is like what happens when the baby is born. So this happens in neonates, right? When the babies are very, very young. So they are newly born. So what happens because of the arterial ischemic stroke, these babies, they starts having seizures. And these seizures are like very, very severe. And it is like very difficult to control. So because these are babies, we cannot do a lot of anti-seizure drugs. So that's why the first line of therapy is phenobarbital. Everybody must have heard of phenobarbital. But what happens? What is the problem here? That because these babies cannot be given like a very high dose or a different drug, there is typically refractoriness from phenobarbital. So most of these babies, they do not respond. Why they do not respond? Because there are different mechanisms of resistance that happens. Okay, so phenobarbital, it mainly works using it enhances the effect of GABA. We all know that it opens the GABA channel for a prolonged period of time, and because of that, it allows the chloride ions to go inside the neurons. And that's how it hyperpolarizes the neuron, and neuron become less excitable and do not seize. So it suppresses the seizure activity. That is how it typically works. But what happens over the time, I have a very nice schematic for this. What happens over the time that these chloride ions, they are not extruded out of the neuron. So they stay inside the neuron. So because they stay inside the neurons, the neuron stays there is like hyperpolarization, and there are more and more seizures, and the baby continue to seize. So there is a difference in GABA activity between a neuron of a child and our neuron. So here you see in a neuron of an immature neuron, like a neuron of a newborn, GABA is not inhibitory. It is excitatory. And as soon as the neuron matures, what happens? The GABA become inhibitory. So this is like a very, very basic thing that happens with the neuron. Typically, GABA is inhibitory. This is what we are reading in pharmacology since forever. But in immature neurons, GABA causes depolarization, and it is excitatory before the developmental shift happen in babies. Okay. So in normal brain, what happens? GABA leads to entry of chloride. You are seeing here. Let me try to mark it. So chloride is entering the cell, okay, because of GABA. But what happens in case of this disease, that I'm adding phenobarbital here. So it is extruding the chloride. Chloride is going outside, right? But because of that, but there is another transporter which is in picture. It is called KCC2. So it is also making chloride go out. But what happens in this disease in case of when there is resistance, this doesn't work. Okay, this track B down regulates KCC2. So what happens? Chloride stays in, and the baby keeps seizing. So the neurons are very hyperexcited. So what I did, in addition to phenobarbital, I inhibited this also. So what happened because of that? It inhibited GABA depolarization, and we expected that it will protect against seizures. So this is like a mechanistic permutation and combination I was doing, that, okay, what will happen if I block multiple targets instead of one target? Like I discussed that in drug discovery, you need to play with the receptors and you need to see that, okay, if this is going up, what else can I inhibit that it will go down again? So I did the same thing. So I, instead of using only phenobarbital, I was using combination therapy. So for that, so I wanted to use one animal model, right? As we are talking about animal models here. So this is my animal model that I was using. So you can see this is like a baby mouse, right? This is like a PND7, 7-day-old baby mouse. So I ligated the carotid artery. So carotid supply blood to brain, okay? And I tied it. So if I tied it, blood will not reach brain and there will be ischemia. This is very basic. We are studying this in pharmacology since forever in anatomy physiology, that if blood oxygen is not supplied to a particular area, there is an infarct and ischemia happens. So same thing is happening here, nothing very fancy. Now I wanted to record the brain activity also. So I implanted electrodes. This is the head of the mouse. Okay, so I implanted 1, 2, 3 electrodes here and I recorded EEG, electroencephalography, to see how much my mouse is seizing. And then I recorded a baseline for 60 minutes with the seizing mouse. And then I gave the drug and then I recorded again to see if there is any decrease in seizures. So because here maybe everybody is not familiar with seizures, I wanted to show this. This is like a basic EEG trace you see when you want to record the brain activity. In very basic language, I can tell that. So you see here in A, there is like a very flat, very flat signal. And then suddenly you see this increase in activity like this, right? So this is what happens when there are seizures. So these are called spikes. So whenever there is electrical activity in the brain, you see a spike on EEG. So here in B, you are seeing so many spikes. And when the patient is actively seizing, you see so many epileptic seizures. Like in the D, you are seeing so many of them. So we observed that when I tie carotid, these animals, they also start seizing like that. Very actively, they were seizing. And then I wanted to check the electrical activity, whether it goes down with my treatment or not. Like, because here I discussed that I blocked this and I also blocked this. So I was playing with receptors. And then I looked at, so this is just for your knowledge, that how you differentiate a spike. So for example, this is like a normal EEG, and then suddenly you see this kind of activity. So here, this is where you know that, okay, there is a electrical signal in the brain and it should actually disrupt the normal morphology. So it should disrupt the baseline and it should be very blunt. And as you see, like here, it is a lot higher than the normal baseline. It goes, suddenly it goes down and goes up. So like that, you basically see when there is electrical activity in brain. I hope I am able to convey this properly. And when we implanted electrode and we tried to see that, we saw that with phenobarbital, the electrical activity did not go down. But when I did a combination therapy, my scissor burden, the electrical activity, it went down. See, from 68 to 85% with combination therapy. So only with phenobarbital, it went down 68%. But when I did a combination, I received a better response. So this is the kind of thing that I was hoping. So now I wanted to check other things like a dose response. In drug discovery, what we do, we look for efficacy and then we see like what is the best dose that we can give to the patient. So I tested a number of drugs, number of doses, like 5 milligram per kg, 10 milligram per kg, 20 milligram. You can see in this graph, right? And I found that 10 milligram and 20 milligram were same. So, which means, so if two doses are showing same effect, what will you do? Like from our previous discussion, you will select the dose which is lower but showing a good efficacy. So this is how we decide that lowest efficacious dose with maximum efficacy. So this is how we typically do in drug discovery process. That is why I wanted to show this data. And then so I got to know that, okay, I need to combine this and this drug in this particular dose. But next, are they toxic? If I combine in this high dose, will my drugs be toxic? This was my next question. And then I observed that there was no toxicity in long term. So I allowed the mice to become 21 days old. They were 7-day old when I treated them. When I recorded the electrical activity, I allowed them to grow old. And then when they became juvenile, I did some behavior tests and they were all normal. So everything was normal. You see, there is no difference. So this is like a heat map of the open field. So mice were just allowed to move around and see, I looked at their activity. And if the brain, I looked at the brain size also. So everything was normal. So no toxicity was observed. So what you will do when you will look for a drug discovery kind of thing, you will try to see if a drug is showing efficacy. If not, we will go for combination therapy. Then we will look for most efficacious dose with the least toxicity. This is what I tried to do. And then I also wanted to see if the neurons are degenerating at that dose. So here, this is like a brain section, coronal section. So I took the brain from the mouse and I cut it. And then I stained it with fluorogate C. So it is a stain which we typically use for degenerating neurons. So when neurons are dying, they take up fluorogate C and they are stained green. So this here in this picture, you will see a lot of green and blue, right? So blue is the nucleus, and wherever little bit you see green is fluorogate staining. So we saw that very less neurons were dying. And we looked at it at different brain regions like ACA, MCA, like anterior cerebral artery, medial cerebral artery, hippocampus, striatum, like thalamus, a lot of brain regions I looked separately. I wanted to see the efficacy. Now I looked at other doses also. So this is like I also looked at low dose because I wanted to go for lowest efficacious dose. So I reduced the dose and tested it again. But this time I did not see efficacy. So I was sure that the dose I used previously in combination was the best dose. So I went ahead with it and I just optimized this kind of therapy, this kind of combination therapy, which where we looked at anti-seizure responses. We did a dose response. We also tested for toxicity, and then we also reduced the neuronal cell death. And then we tried to look for other doses also, but we did not find a good response. So this is like a basic of how I did some of my work. This work is from my initial days of postdoc when I was at Kennedy Krieger Institute. It is a children's hospital. And I think this is it. So if you have like any question or if you want to ask anything, I'm very open to the discussion. Or I can also talk more about some cell culture models if we have time.
Yeah, is there any questions you can ask the Dr. Preeti Vyas ma'am? Ma'am was ready to give the answers. I think it was clearly explained, ma'am. So no one has any questions for it. So,
Okay.
okay, ma'am. Thank you, ma'am, Dr. Preeti Vyas, ma'am, for your inspiring and informative lecture. On behalf of VV Institute of Pharmaceutical Sciences, we express our heartfelt gratitude and present a token of appreciation in the form of a certificate, ma'am.
Thank you so much. It was great to present
Yeah,
with everyone here.
yeah. This is a
Yeah.
certificate, ma'am. Thank you, ma'am.
Good afternoon, ma'am. Preeti madam, good afternoon.
Good
Hello,
afternoon.
I'm audible. Yeah,
Yeah,
ma'am,
yeah,
this is Dr. Anand.
you are.
Yeah, this is Dr. Anand Chaurasia from Bhopal, MP. Actually,
Hello,
ma'am,
sir.
I'm working. Yeah, I'm working as a director of academics and principal. Actually, my question is that there is any biomarkers for the neurotoxicity is available for the preclinical estimation of the given some project of the students. Is there any biomarker available which could be feasible
For for toxic.
economically also? Yeah, for
Oh,
neurotoxicity, or for
so
the any neurodegeneration based biomarkers like in Parkinsonism or in Alzheimer's.
so are you asking for cell culture biomarkers or in animals? I can explain
Or or in,
both.
or in animals animals only.
Animals only. Okay, so
Yeah.
sir, how we were doing or how I typically do tox studies is that you do like a once you have your dose for efficacy, you do like a 10 times or 20 times higher dose, or like you can select, you can go like 10 times higher, 50 times higher, 100 times higher, that kind of dose. And then you collect all the organs from the animal after some time. Or you can do like a chronic dose study. You can inject high dose for a long period of time and take all the organs from the animal, like heart, liver, kidney, lungs, brain, everything you take. Okay. And then the cheapest way, the most economical way to do is to do like maybe an H&E stain and see if you see dead cells in the whole body. Also, you can do like a caspase 3 assay, LDH release assay. That kind of assays are good. You can look at LDH leakage or
Ma'am, please repeat again. Please repeat again. First one is that uh,
first one, you
before
can
to
do
catch
either
test.
like a you can do like a acute toxicity study or maybe a chronic
Yeah.
toxicity study where you inject like a high dose in animal and then you collect all the organs and do H&E staining. So this is
ठीक
very
है.
economical. Okay. And then
And
what you can
second
do,
biomarker.
second biomarker is you can take the brain and then is there a specific area? What is the disease you are working on? Is it Alzheimer, Parkinson?
Both are
Both.
neurodegenerative disease. Any neurodegenerative diseases. Common
So
like that.
for Alzheimer's, I would say if you can micro dissect the hippocampus, or you can do the whole brain also, but if you can micro dissect the hippocampus or substantia nigra, take that part of the brain and measure caspase 3 and LDH, lactate dehydrogenase and caspase 3, and you can take like an annexin kit. So these are not very expensive.
So
You
is
can
that the
do.
distinguished that it's supposed to we have to retrieve the brain? So is that the
Mhm.
any particular that part should be very means
Yeah.
uh, easy to trace that part only because I think so if you are not expertise it could not be able to see the proper part of the neurons.
Yeah,
ये लगता है
maybe
sir.
no, maybe you won't be able to isolate substantia nigra, but hippocampus is pretty easy. Or you can do like you can remove the cerebellum and you can do the whole brain. If you think that okay, I
Okay.
cannot micro dissect all the areas, you can do the whole brain, and you can measure LDH. You can measure the markers for apoptosis, necrosis, ferroptosis. So these are very cheap. And in the liver also, for
Okay.
liver, if you are testing your drug and you want to look at liver toxicity, you can buy those AST and all those kits, and they are also very cheap. The human LDH kit you can buy, it is not very expensive, and you can use that. We were using it in India DO, so it is very affordable.
Okay, ma'am. Very nice to interact you. And second thing, ma'am, if share please your contact number, WhatsApp or email for future any reference or contact. It's supposed to
Oh
be
yeah,
there.
I ghmi.edu. Okay, I typed my email in the comment section in the meeting chat. So if anybody want to contact, I would be happy to contact with you. Naharika has my contact details in case.
Yeah, madam, you can share on that
Yes,
FDP WhatsApp group. So
yes,
we
sir.
we
Yes,
will
sir. I
get
will
it.
share. Okay, sir.
हाँ, उससे हम
Okay,
लोग
sir.
निकाल लेंगे मैम. कभी कोई मैडम से बात करना होती है, कभी कुछ ऐसा रेफरेंस, फ्यूचर रेफरेंस, we
Okay,
will do
sir.
that.
Okay, sir. We will be in touch. Okay, ma'am.
Anyhow,
Thank you,
it's
ma'am.
very nice introduction.
Thank you so much.
Thank you for your support, ma'am. Though it's night 1 a.m., you are here to
Oh,
spend the time.
it is
Thank
now
you.
3 a.m.
Oh, okay, ma'am. So here is
Thank you so much. Can I exit?
ma'am, it's the token of our appreciation, ma'am. Thank you, ma'am, for
So
your
much,
valuable
Narika.
spending valuable time. Thank you, ma'am.
Thank you so much.
Okay, ma'am, you can exit, ma'am.
Thank you, everyone. Thank you for having me here.
Thank you, ma'am.
So, ma'am, please share the
Oh.
feedback link on the WhatsApp group so we can submit our feedback.
Dear participants, the attendance form is posted in the chat box. So everyone make sure to please fill out the form before leaving the section. So the attendance is mandatory to get the e-certificate. So I request everyone to kindly fill the Google Forms for day 1 attendance. It was posted in our chat box, and again it won't be posted in WhatsApp group.
Okay.
So before leaving
Excuse
the
me, ma'am.
section, yes, ma'am.
Ma'am, actually signature column upload చేయడానికి రావట్లేదు మ్యామ్ అది.
It's in JPG
ఎంబీ
format లో మ్యామ్.
ఎక్కువ
మీరు
ఉందని చెప్పి రిజెక్ట్ చేస్తుంది.
దాన్ని తగ్గించుకొని అప్లోడ్ చేయాలి మ్యామ్. 1 MBకి కన్సైజ్ చేసి
అది తెలుసు
గూగుల్లో
మ్యామ్. అంటే అంత మామూలుగా మన దగ్గర ఉన్నది పెట్టేస్తాం కదా.
అంటే అన్ని ఈక్వల్ గా ఉండాలి కదా మ్యామ్. ఎవరికి నచ్చినట్టు వాళ్ళు పెడితే మళ్ళీ అవ్వదు కదా మ్యామ్. సో 1 MBకి కన్సైజ్ చేసి అప్లోడ్ చేయండి మ్యామ్.
लोकल लैंग्वेज बोलने लग गए.
Ma'am, for each signature, that how could we? Hello. Wow.